Hello.
Hey, Trevor. Click on the wrong one. No. Oh, better. No. What's going on there? Oh, what are you doing, my friend?
Hello.
There it is. Weird. That code should work. Trevor, you can hear me okay, right?
Yeah, I can.
And now we can hear you. You are on a speaker in the back of the room. So hopefully, if this will work. Oh, we got one more person. Hold on, let's get Gabby in here too. We can actually say hello to each other without me. We're gonna find out. All right, Trevor, Gabby, say hello to the 5 people in the classroom right now.
Hello, 5 people.
Oh, Gabby, we can't hear you.
Yeah, it's a bit laggy, but hi.
Hi. Okay, now see if you can say hi to them. People in the classroom. Can you hear that?
Yeah.
Okay.
Yeah.
All right. Gotta bring us together somehow. This is crazy. This is still crazy, right? Yeah. Okay. I'm trying. All right. My hope is you can see the code. I don't know if it's giving you the option to select our class from the drop down by default, or if I have to invite you every time. But regardless, the code should be there. And here comes Miguel from the car. Hey, Miguel, we can actually hear you now in the classroom. Everybody can hear you are on a speaker.
Oh, my bad.
Say hi.
What's up? Here's Pablo. Hey.
What's the
Good morning.
He's attending class from the car.
I'll be there in 1 second.
You would not be the first person. All right. Shall we get started? Keep logging into those Pear Deck slides to the best of your ability. Okay. Want to do our check-in. I thought it might be especially important to our check-in today because this is yet another transition in our lives, right? A transition that now is what, 4 days old, but really what, 2 days old, right? Because you just kind of flip-flopped each day this week. So this one is called I Miss I Have. We actually may have done this in the past, but I wanted to start there. And then really we're going to spend the meat of class today telling the story of this scientific paper, this Stinson et al. Paper. So my hope is you had a chance to look at it. You know, in normal times, this would not have been the first scientific paper that we would have read together. But it's important to note that if you were challenged by this, that's 100% normal. Okay? This paper is not written for high school students. It's written for other scientists. So you may have encountered jargon, right? Scientific vocabulary with which you were unfamiliar. My hope is you looked it up, right? But if you didn't, I want to give you a time here in a moment to ask questions, okay? But ideally, a good scientific paper tells a story. So this morning is story time with Mr. Bergeron. I want to try to tell you the story of this paper. Chapter by chapter, page by page, all 4 of them. Okay. And then we'll wrap up things with a quick exit slip because you got a text a couple days ago giving you access to the 4th take-home assessment. Okay. So I want to make sure if there's anything that we can do between now and Monday that we address that. Okay. Folks at home, can you still hear me okay? I took my earbuds out, so I hope the microphone's picking me up all right. What questions do you have for me or for the good of the group before we get started? Anybody in here have a question? You can still put things in the chat. I apologize if I walk away from the camera, but hopefully you can still hear me no matter what. All right, well, let's get started with our check-in then. Again, another period of, our district calls this another pivot. I often wonder when we're going to pivot back, especially given what's happening in our local area right now. But I don't know the answer to that question. So in this period of transition, in this period of change, I wanted to give you an opportunity to pause, to reflect, and just jot down, is there something that you miss? It could be something from last week. It could be something from 6 months ago. Okay. But then also, could you refocus your effort, your attention on identifying something that you have right now for which you are grateful? Sometimes reframing our thought processes through a lens of gratitude can be very helpful during very challenging times. So I invite you to jot down your thinking, your misses, your haves, just in these first couple minutes of class today. We lost Miguel. Oh, no, we didn't. That's why Miguel was in the car. Miguel is here. You can sit where you like. You can sit where you like. I take a picture, so. That makes sense now. Hey, Ben, welcome. You can see the code in the upper right-hand corner. We're just doing our check-in. You haven't missed very much. Glad you're here.
Yep, I got it. Thanks.
So keep writing. I'm just going to kind of try to share what I'm seeing on my end. Not identifying anybody along the way, of course. This is still anonymous. Yeah. So many things are different now, aren't they? Yeah. Of course, there's all those things that we have that oftentimes we take for granted unless we take that opportunity to remember. Day is short. For you folks, I teach this class, this lesson 6 times today. It is exhausting.
You have any classes every day?
Not this year, no. So I have two of these classes on A days and then 6 on B days.
I'm sorry you have to go through that.
Well, something I was going to share with all of you too, you know, something I missed that I didn't even realize I truly missed until Monday was a full classroom of students, right? because you folks give me energy. And when there's 3 or 4 people in the room, like there were on Monday, by the end of the day, I was exhausted. And then Tuesday, when I had 6 classes, I was 3 times that level of exhaustion. So this is tough for teachers too. Please know that. Any teacher who says that it's not tough, I'm going to question their sanity right about now. Yeah, what happened to the daylight? I think I was heading home at 4:25 yesterday. It was basically dark. Yeah, just seeing people, right? Somebody said earlier, yesterday, in fact, they said, I miss my M to Z friends. And I'm like, oh yeah, there's an entire group of human beings that you don't interact with in school anymore. I thought that was a really interesting way to phrase it, though. I miss my M to Z friends.
It's a weird cutoff because it's not the middle of the alphabet.
Well, yeah, and I can assure you that like when I printed out the roster to kind of keep track of who was going to be here and who wasn't going to be here, it's a very arbitrary cutoff because it's not half and half in those classes. Like teachers are being asked to supervise classrooms for those teachers who are still teaching from home. There's a class that I supervise on Monday mornings, 4th hour. There's 1 person in the room and there's 1 person on Zoom. It's a 2-person class. So, and that's just the way the alphabet fell.
Does the other one have like 16 people?
No, it's still overall a pretty small class, but in the afternoon, the PM session, right, there were 6 people in person, and then I think 2 people online.
So why don't they just move they move the letter until it's half. Okay, then.
Sorry, folks at home, I wish you could see I'm doing a little dance because if you can hear what Miguel's saying, why don't they just set it up so that it's half and half? Logistically, what do, right? One teacher has this half of the alphabet, right? And another teacher has the other half of the alphabet. You communicate that across 6 different teachers. I mean, it could,
Oh, that's true.
you'd have to reconfigure the schedules, right, for every single student potentially, based on the way the alphabetical list fell for each teacher, for each period.
Yeah, but like if there's a general trend that there's less, like couldn't you move like at least like the M's and the N's? Maybe that helps a little.
No, I think these are all good suggestions. All right. Well, thank you for sharing your misses and your haves. Whoops. You know, for me, I'm still grateful that my 2 3-year-olds, 3.5-year-olds now are mostly blissfully unaware of what's going on in the world. Yeah, they're missing out on preschool, but they're not missing out on kindergarten. Those are them, by the way. Little age pictures. I need to update them, but and my family is mostly happy and healthy. Well, maybe not happy, but healthy. All right. Well, I'm hoping you can see the next slide. We want to transition into story time based around this scientific paper. And it's a tough read the first time you read a scientific paper. You know, it doesn't look very long. It's only 4 pages long, but there's a lot going on there. So I want to give you a chance to pose questions that you have about the paper. So if you'll just take a few minutes, as you think back on what you read, what are you still uncertain about? What do you have questions about? This might be a good place for us to start. Start story time. You can type them right in the box. If you want to ask them out loud, you're welcome to. I just thought one spot we could capture everything would be on this slide. always welcome to ask questions out loud. So that it can be hard for people at home to do that. I don't hear any keys clacking any longer, so yeah, we definitely want to think about the role of this invasive garlic mustard species, right? And how it's disrupting this ecological interaction. You know, that really is the connection to what we did the other day, right? It's all well and good to study these ecological interactions and identify which type of interaction is which, but these interactions are real things. And there's a well-established interaction between these fungi and the roots of these woody trees and native forest ecosystems. But upon the introduction of this garlic mustard plant, lo and behold, the garlic mustard plant appears to have the ability to disrupt that mutualistic interaction. And one of the things I hope to share with you today is that there are both short-term and long-term consequences to that disruption. So much so that we might even argue that garlic mustard plants are long-term planters. Not long-term planters, long-term planners. They are setting up a system that promotes their long-term survival and reproductive success over and above all of the other species that might be present within the system. In many ways, they're using natural selection to their advantage by disrupting these interactions. Yeah, the experiments do seem redundant, but hopefully the story will reveal how they're subtly different and how chapter by chapter we get a little bit more information about how garlic mustard is exerting its effect. So the plants themselves aren't being sterilized, but the soil is being sterilized. That's an important point that we want to highlight. Yeah, we're definitely going to spend some time on the figures. Materials and methods, they are by convention included at the end of a scientific paper. Yeah, and it's only 4 pages, but there's a lot going on here. There's a lot going on here. So it looks like are we ready to kind of dive in? So you keep asking those questions. Again, people at home, we can hear you. So if you want to take yourself off mute, it's like you're kind of raising your hand and saying what you need to say. And I think people here in the classroom will be able to, you'll be able to hear the people in the classroom if they ask questions as well. Okay, so please don't hesitate to jump in if you need some support. Okay. All right, so I'm on slide 139, and I thought it might be helpful just to kind of begin with what this interaction between the fungus and the woody plants, the tree, right, actually looks like. Because when we think about a fungus, I bet, I don't know, you think about a mushroom, right? And oftentimes we think about like the mushrooms we find at the grocery store, right? Or maybe in our refrigerator. It's like this little, I don't know, toadstool. But in reality, fungi are far more complex than what we see growing above ground. Okay? They're not plants, so they don't have roots per se, but they definitely have these extensions that grow out from the base of the fungus that if you could see them, like in the picture I have on the screen right now, would look like roots. Okay? So what a lot of fungi do, because they're not plants, they are not really animals either, but they're closer to animals than they are to plants. They're acquiring nutrients from their environment. They're eating things. They are heterotrophs. But one of the ways that they make the consumption of nutrients far more efficient is by interacting with the roots of trees. And what they literally do with these extensions is they burrow into the root cells of the plant, and they siphon sugar away from the plant. But it's not a parasitic relationship because there's plenty of sugar to go around. The plant is not being harmed by this interaction. In fact, the interaction is actually beneficial to the plant and the fungus. It is indeed a mutualistic interaction because what that little extension burrowing into the root cells of the plant accomplishes is a more efficient delivery of nutrients, polyatomic ions, nitrates, phosphates, things in the soil that the plant needs to grow and develop over the course of its life. Okay. So that's an important piece of the puzzle. Okay, so as you kind of look from left to right, I tried to create an image that shows big picture, how are the fungi and the woody trees interacting, and then zoomed in, there's of course on the left-hand side of the right image, no interactions between the fungus and the woody plants. And on the right-hand side of the image on the right, you can see those interactions. And these are the AMF interactions, arbuscular mycorrhizal formations, the interactions, physical interactions between the fungus and the plant, right? So just pausing there. What questions does this bring to light for you? Were you aware that under the typical forest floor, there are this level of interactions taking place? Most people aren't, right? That's okay. All right. So this next image, this is garlic mustard. Yes, if you were to eat it, it would taste a little oniony and a little spicy. It's totally edible. And if we see it, we would think that it's a weed. And it grows here in Missouri. It is an invasive species here in Missouri. It does not belong here.
Where does it belong?
It was originally thought to have been brought over from most likely European countries. Exactly when and exactly how is still being debated by scientists. But what we know is that when it arrived in North America, it took advantage of a complete lack of competition in these non-native systems, these non-native ecosystems that allowed it to thrive and flourish. Okay. I included a picture not because at this time of the year will you be able to find any, but I bet, especially those little white flowers, I know I've seen those in my backyard. So there's garlic mustard growing in my backyard. So what is the connection between the garlic mustard, the fungi, and these woody plants? Well, that's where chapter 1 of our story begins. Okay. So can everybody see the figure on their ends here? Excellent. And people at home, you can see the figure. This is figure 1, I hope. Wonderful. Okay. So the story begins with these scientists going out into the world and identifying areas where garlic mustard is growing and garlic mustard isn't growing. And I'm not exaggerating, digging up the soil in those areas and bringing it back to their laboratory. Because what the first question they want to know is there something in the soil that is potentially interfering with this mutualistic interaction between the fungus and the plants? Okay. So they bring that soil back to their labs. And again, I'm not exaggerating. They're going to fill hundreds of cells, not body cells, not plant cells, not animal cells, but little cells where you might grow a plant with that soil. Hundreds of them. So picture an enormous greenhouse. And then into that soil, they're going to put different seedlings. So you've got the white ash seedlings. You've got the, sugar maple and red maple seedlings. They're all going to go into that soil. And they're going to wait 3 months. Yeah, no big deal, just 3 months. And they're going to come back after 3 months and they're going to ask 2 questions. Is the interaction between the fungus and those woody plants interfered with, interrupted, based on whether or not these plants are growing in the soil where the garlic mustard was present or the soil where the garlic mustard wasn't present? As we look at the figure, so I'm in figure 1A up top, and it can be a little tricky because the labels for each of those bars, they didn't put them on both graphs, they just put them on the bottom graph. But all the labels for figure 1B are the same labels for figure 1A. As you move across, so invaded, uninvaded. So Ashna, as you look at that, just that first part, figure 1A, invaded versus uninvaded, in respect to percent mycorrhizal colonization. What claim do you think the scientists could make? Sorry? Okay. If the soil that they brought back to their labs had been invaded by the garlic mustard before they brought it back to their labs, I can't hear you, I'm sorry.
The garlic mustard had already grown there.
So the garlic mustard was growing out in the wild, right? They dug up the soil where the garlic mustard was either growing or where it wasn't growing and brought that soil back to their labs.
Yeah.
And then grew these seedlings, these tree seedlings in that soil. If you look at figure 1A, I'm just on the invaded versus uninvaded part, right? How are the plants, or how is, in this first part, the mycorrhizal colonization? How is the fungus, mycorrhizal is another word for the fungus, how well are they able to colonize the plants, the roots of the plants, if they were growing in the invaded soil? Horribly, right? Some of the plants aren't forming any interactions. What are those? The sugar maple and the white ash, right? And then the red maple is barely forming some interactions, what, about 10% of the time, give or take? Actually, probably less than that, 5% of the time. Compared to the uninvaded soil, where the garlic mustard was never present, how are those plants doing? And with those error bars, it's a statistically significant difference, right? They are doing significantly better in the uninvaded soil compared to the invaded soil. When we say they, we're still referring to the interactions between the fungus and the plants.
So is this like an indirect relationship between the like garlic mustard and the trees? Is it more like how it affects the fungus and then how that affects the trees?
So it's a really important question that Jeremy's asking, right? Right now in chapter 1 of this story, right, we are definitely only able to make the claim that this effect is indirect, right? Because it's literally something in the soil where the garlic mustard plants were growing. The garlic mustard plants aren't even there anymore in the lab, right? It's just the soil. That's all. So if they're having an effect, they don't even have to be there to have the effect on this interaction.
What is the sterile soil?
That's the next step, right? Okay. So actually, before we get to the sterile soil and the kind of the right-hand side of figure 1, if we go down to figure 1B, now our measurement, right, on the y-axis is percent increase in plant biomass. Really, it's a measurement of how much the plants have grown, right? How are the plants growing? How well are the plants growing, Annabelle, in the soil that was invaded by the garlic mustard?
Mr. Bergeron, the pear deck is still on the picture of like
Thank you.
Thank you.
Thank you.
Thank you.
How are the plants growing in the invaded soil? In figure 1B on the left-hand side. Right. I mean, we would argue the same thing, right? They're barely growing at all. What is that? Less than 10% in respect to their increase in biomass. And biomass is literally that, right? The increase in living material. So new leaves, new stems, new roots, things like that. Okay? They're barely growing in the invaded soil, but in the uninvaded soil, they're doing okay. They're doing okay. So those connections, right, those interactions between the fungus and the plant have been disrupted in the invaded soil, and the plants aren't growing very well in the soil that had been invaded by garlic mustard. So we go to the sterile soil. They're sterilizing the soil, right? Why the heck would you sterilize the soil? Well, first off, to sterilize soil is not as hard as it sounds. We actually have a machine in the back behind Annabelle where you could do something like that. You can't see it. It's in the back. It's called an autoclave. It, in essence, just heats things, anything, to about 120 degrees Celsius at about 15 psi. So if I were to put you in there, you won't fit. But if I put you in there, you're not going to make it. You're going to die. And so will any of the microorganisms. So would the fungus. living in the soil. So by sterilizing the soil, you eliminate all of the fungus from the soil. Whether it's invaded or uninvaded, it almost doesn't matter, because we can see if I do that, are the plants, well, first, are the plants forming the interaction with the fungus? Duh. Of course not, right? The fungus is dead. And similarly, in respect to the biomass, whether the soil is invaded or uninvaded, is there a significant difference between whether or not the soil was invaded or uninvaded if we sterilize the soil? Those error bars overlap like crazy. Right? So, chapter 1. There's something in the soil that interferes with the interactions between the fungus and the roots of these plants. All right? Questions? Comments? Folks at home, we doing okay? Chapter 2. Now, this experiment's a little bit different. Now they're not going to worry about field soil, right? Soil that was out in the world where either garlic mustard was growing or wasn't growing. They're going to use their own soil. And they're going to condition that soil with the different types of plants that they're trying to grow. So they're going to have their soil. Here, I got a picture of this too. And Ben, remind me to go back if I forget again. They're going to have their soil now, like soil you would buy at the soil store. They're going to fill all those cells of their tray with that soil. And now they're going to grow garlic mustard, red maple, sugar maple, or white ash plants in that soil for 3 months. They're going to condition the soil with those plants. Then they're going to pull out all those plants. And they're going to now add back garlic mustard, white ash, sugar maple, and red maple plants to that conditioned soil. Right? So soil where those plants were growing before. Can you picture this? Our greenhouse has got a lot bigger. Okay. And then they ask the same question as before, right? Okay. If I look at, so now I'm in figure 2, figure 2A, right? If I assess that percent mycorrhizal colonization, again, the interaction between the fungus and the plants, in which type of conditioned soil does that effect go away? What would you say?
I guess, based on what I can see on there.
Oh, sorry, let me go back. Thank you. Daniel, remember, you're supposed to remind me.
All of them like are pretty similar except for when they're put into the garlic mustard plant soil.
It's only the soil that had been conditioned with the garlic mustard plants that had an effect on the ability of the fungus to colonize these woody plants. If you grow the woody plants in sugar maple soil, or white ash conditioned soil, or red maple conditioned soil, doesn't matter. No effect. But if you grow those plants in the garlic mustard conditioned soil, there's no interactions that are forming. And moreover, Jack, what we say about the percent increase in biomass? Which type of conditioned soil reduces the biomass the most? The garlic mustard. Okay. Plants don't like to grow. These plants don't like to grow in the presence of garlic mustard. Okay. Yeah, Miguel.
So if you look at the things under where it says figure 2, it says that the degrees of freedom are 449. What does that mean?
This is a different type of statistical test. You've never seen
Oh, okay.
this statistical test before. That's why it looks unfamiliar to you. But in essence, just quickly to summarize, by degrees of freedom, they're referencing, can you see across the x-axis, there's 5 different groups?
Yeah.
So think of degrees of freedom, 5 minus 1, that's where the 4 is coming from.
Yeah.
And the 49 is coming from the number of replicates. So the tray, right, that I tried to create that image of, you've got 49 plants of each type being grown. And that's where all of these data are coming from.
Whatever. What do you have 50 and it would be 50 minus 1 or is it just 49?
In that case, it is actually 50 minutes. But that level of detail is beyond the scope of our class. But it's still a good question. All right? All right. So I think for the sake of time, we're going to go to figure 3. Because this is my favorite figure by far. Right? Jeremy asked about, like, is this effect, all going all the way back to figure 1, is this effect indirect? Yeah, it's indirect. But how is it indirect? So in figure 3, the scientists take the garlic mustard plants, right? The white ash, sugar maple, the red maple plants. And they, I'm not kidding, they grind them up in a blender with some water. They make an extract of the garlic mustard plants, the white ash plants, the red maple plants, sugar maple plants. And they add that extract to the soil where these different types of plants are growing. To ask the question, are the garlic mustard plants producing something chemical that's interfering with the ability of the mutualistic interaction to form and thus decrease the plant's ability to grow and develop over the course of their life? I mean, I'm a nerd, folks. I'm a huge nerd. The plant is producing a chemical which gets released into the soil, figure 1 and figure 2, that directly, or shall we say indirectly, impacts the ability of the fungus to interact with the roots of these plants. What is figure 3 telling us? Probably which extract has the most significant effect on the ability. You can take it A, B, or C, right?
No, wait, no, sorry.
Which extract has the most significant effect on either mycorrhizal colonization, increase in plant biomass, or if you want to go to the level of percent spore germination, you could. But which type of extract has the most significant effect? Up now.
The garlic mustard.
Down below, right? GM extract, garlic mustard extract. For the most part, the other extracts that have been added to the soil where these plants are growing, you're not having a significant effect. But if you add the garlic mustard extract, you do. There's something in the plant that impairs the ability of the interaction to form and for the plants to grow.
Wait. Add. So looking at the second graph, the increase in percent
Mhm.
biomass.
Mhm.
So when the white ash extract was added to the white ash plant,
Mhm.
it didn't grow as much compared to when other extracts were added,
Mhm.
ignoring the GM. And the same thing for the RM
Mhm.
for the red maple. So when you add like the plant's own extract to itself, it doesn't do anything.
Probably not, right? Because we think about these plants, these trees, in this case, growing next to each other. Even if they were releasing a chemical into the soil, those plants probably don't really care if they're growing close to one another, as long as they're not growing too close to one another. So I think what I heard you say at the beginning, but maybe you corrected yourself mid-sentence, was, could there be an effect of one type of extract on the same type of plant?
Yeah.
Absolutely there can be. So if I'm a plant of one species and I'm growing too close to another plant of the same species, a signal being sent from one plant to the other that says, stop, you're too close to me, could be a really powerful signal. Because by growing too close, you might begin to compete for resources that can make it harder for both of you to survive or reproduce. And that happens in nature all the time. But in this case, it's the garlic mustard plant that has the most significant effect. There's something in these plants that disrupts the interaction. So figure 3 is my favorite part. Figure 4 is a close second. Because in figure 4, what the investigators are beginning to argue is that, wait a minute. Could this effect of garlic mustard be bigger than just the effect on the white ash, the red maple, and the sugar maple? What they begin to argue for is if I'm a plant that's dependent upon an interaction with a fungus, right, to gain nutrients, the more dependent I am on that interaction, the more I'm going to be affected if garlic mustard is also present in the environment. Now we have to be honest, we're almost out of time, so I'm gonna go quickly through this last part. But the effect, right, the relationship, the correlation, not the strongest in the whole wide world, right? R squareds of 0.1 for figure 4a and 0.415 for figure 4b. They're not super strong correlations. They may not even be statistically significant, right? But I think we could argue that there is a relationship-ish, which is not a very strong one. Right? But here's why that matters. Because I mentioned at the beginning of class today that these garlic mustard plants might very well be long-term planters. So here, there's evidence that they're producing a chemical that moves into the soil that has an effect on woody plants. So I like this picture because you can see the garlic mustard on the forest floor. That's those small green plants. But you can also see the trunks of the woody trees in the image. It's a little bit harder to see, but can you see or can you imagine the canopy of leaves and branches that would be at the top of the forest? The garlic mustard plants are producing a chemical which makes it harder for those woody plants to obtain nutrients, to grow and develop over the course of their lifetime. I'm thinking tall trees first, big trees, old trees first. But it's also going to impair the ability of baby trees to grow and develop over the course of their lifetime. So how might you predict the forest itself could change as a consequence of the garlic mustard invasion? How might the canopy change?
Might become lower.
Light's still hitting the ground when the canopy is intact, right? But if somehow the garlic mustard plant could impair the ability of these woody plants to form that canopy, maybe, just maybe, what we would begin to see, is something like this. Trees start to die. The canopy becomes more permeable. More light can now strike the ground than before because the canopy's been disrupted. Folks, the garlic mustard plants have altered the environment in a way to promote not just their short-term success, but their long-term success moving forward. They are long-term planners. Does that affect bottom up or top down? Remember, they're producers, they're plants. Bottom up.
Bottom up.
But this time, the bottom-up trophic cascade, it's not helping higher trophic levels, it's harming higher trophic levels. That's how it all connects.
Aren't they both producers?
They are,
So are they
but
on the same trophic level?
they're on the same trophic level, but remember, these plants, they're invasive, right? They're non-native, they do not belong there. It's almost like in the lab 7, if you introduce a new plant species, in this case, the garlic mustard, you can still have a bottom-up cascade, right? Because we're at the level of the producers in this case. That could promote success, but in this case, does not. All right, I hope story time helped. They close their laptops here in the classroom, but if you folks want to add something in respect to what you need before you sit for the assessment, before you take the assessment, I would welcome feedback. But if you don't have any more,
Thanks.
Hi, Mr. Bridger.
see you later.
Thank you. Bye.
Bye.
Thank you. Thank you.
Last class.
Thank you.
You're welcome.
Do I have to spray it down?
I have to spray down.
Oh.
So yeah, sorry. I was wrapping up this last class and you just happened to be the first one in the room. If it's dry, no one will sit there.
Well, we're gonna do next.
Yes, actually. Only 5 people here. It's dry, nobody's back their last period, I'm glad somebody did. Hey, Sarah. Have any interest, you can actually now say hi to people in the classroom. They can hear you through a speaker. That is not a mandate. That is an opportunity. But Maya's not here yet. I thought maybe you might want to say hi to Maya. Code is up there. I need the code. Drop down menu though. We can pick our class. All right, so as everybody logs in, I'm trying something new, folks at home and folks here in the classroom. There is a speaker behind Sasha that we can use for people at home to, that we can use to hear the people at home, but hopefully they can still hear us via the microphone. So I thought, if you're willing, we'll start with the people here in the classroom. Let's just say hi to the people at home. Ready? Count of 3. 1, 2, 3. Hi, everybody.
My paradigm isn't working.
Okay, we'll get it up in a second. All right, so people at home, if you're willing. Oh, wait, I got 2 more people who are trying to join. All right, people at home, if you're willing, take yourself off mute and say hi to everybody here in the classroom.
Hello.
Hi.
They can hear us.
They can hear you, and now we can hear them.
I can speak now.
Wait, is that a good thing or a bad thing?
Both.
Keep like wanting to look fast to like see.
I know, right? I think that was Ben at the last one. Yeah.
Yes, it was.
Anybody else want to say hi real quick before we get started?
I said, how did you get that? Did Adam do it?
Yeah, Adam just is.
I sent that to Adam.
It's a fun.
All right. I'm trying to build some semblance of community, right? Even though we're in different places. But this is still weird, right? All right. So agenda-wise, I thought once again, we can start with a brief check-in. This one, I think we've represented before, it's called I Miss I Have. Once again, another period of transition in our lives. So I thought taking a moment to reflect on even maybe over the last couple of days, what is it that you miss? But also taking a moment and reflecting and maybe being grateful for what you still have would be helpful. That's helpful for me. And then really we're going to spend most of the time today in what I'm going to call story time with Mr. Bergeron. I'm hoping you get a chance to look at the scientific paper. Okay. Give it a once over. This would not have been the first paper that we would have read as a group during normal times. But it is what it is. If you found it challenging, that's absolutely okay. This paper is not written for high school students. This paper is written for scientists, professional scientists. So you may have encountered a lot of terminology with which you were unfamiliar. My hope is you looked it up. If not, let's tackle that today. But ultimately, a good scientific paper tells a story. So, story time with Mr. Bergeron. I want to try to tell the story of this paper, okay, so that you get a better understanding not only of how it connects to what we have been doing, but also why it matters for biologists. So, and my hope is at the end we'll have a moment to do a quick exit slip. What questions do you folks, whether you're at home or you're here in the room, have for me or for the good of the group? Remember, you can ask out loud and we can hear you at home. Well, then we'll get started. If questions pop up, don't hesitate to ask. Chat still works too. Again, this check-in is an opportunity to acknowledge what it is that you might be missing right now, but also to acknowledge something, anything, all of the things for which you are grateful. So if you take a moment, maybe even pause, take a deep breath, ground yourself. And then think about what it is that you're missing, but also what you have. This is crazy. Tostitos, the super salty ones?
What?
Are Tostitos the super salty chips, tortilla chips?
Yeah.
It's a good chip. I like the drawing. In bed till 9, but I get to go home at 12 is what I'm assuming that represents.
Yes, that is.
Oh, thank you for sharing.
My artistic ability is professional.
Well, I don't know that one could be an expert artist with the Pear Deck drawing tool, so I appreciate the effort nonetheless. Yeah. Yeah, a lot of the in-person stuff, right? Speech and debate. I heard from some other speech and debaters yesterday that CFC, right? Yeah, it's going to be all virtual, aside from like a, I guess like a mission control sort of type of thing here in the building. And Mr. Sywell and I guess maybe the veterans will be running. Yeah. Yesterday, I asked the same question to the first period students. Somebody made a really, I don't know, it just struck me. I wanted to share it with you too. They said, I miss my M to Z friends. I was like, oh yeah. There's a whole group of people that you still can't interact with based on the alphabet.
Yeah.
I don't know. I thought that was really
We
interesting.
should do it like last name every other, and then like first name every other day.
Yeah.
I mean, it would be really confusing, but it would be
Yeah. Yeah, I mean, yeah. I mean, I can only share like the roster, right, that I use. I just kind of drew a line at L and said, all right, here's who's going to be here in the morning, who's going to be here in the afternoon. But that is imperfectly perfect. You know, you may know that some of your teachers have been asked to supervise classes for those teachers who are still teaching from home. And I do that on A days, so yesterday. And there's a teacher who has, I'm not exaggerating, one student here in school and one student on Zoom. 2 people.
Oh, that's
This
Mr.
one is-
Beecham. That was my class on Monday.
So we're not alone in that, right? So it's not working, right? But sorry, I'm sure the mic picked me up. Maybe people at home heard me first. One thing I tend to do every day is I check the St. Louis County dashboard for the percent positivity rate. And usually it's updated around 10:30, so it's not updated by now. But it was updated for yesterday, this morning. Folks, we're at 19.9%. That is insanity. This virus is everywhere. So regardless of the alphabet, and regardless of our M to Z and A to L friends, I hate to break it to you, this isn't going to last much longer. Because it can't.
My
And the
math
mic
teacher said
picked that
that.
up, and you can post that on YouTube. Please do it.
Okay. My math teacher said the same thing as you, that it's not going to last much longer.
Yeah. So
we're not gonna make it back from break. Like, I know, like, I'm not traveling, but I know there will be people who do, and that will just spike it even more.
sorry, I'm standing up in front of the camera. I apologize. Can you guys hear what Maya said from your end? Good. Yeah. You know, there were those days, and we can't belabor this, but I do want to honor if you have questions, concerns, or comments about what's happening. There were days back in March and April where we were talking about flattening the curve. What in the heck happened? Because I'm looking at a curve right now that ain't flat and is higher or as high as it was back in April. All right. Well, thank you for sharing your misses and your haves. One thing I wanted to share with all of you, I realized it this week, really, for the first time. I miss having a full classroom. You know, on A days, I teach the lesson twice, but on B days like this, I teach this lesson 6 times. And not having a full classroom, I realized you folks give me energy. and I don't have as much of it. So getting through the day is really, really hard. But I'm grateful that my family, for the most part, is healthy. And I hope will continue to be healthy, as I hope your families will also continue to be healthy, and that you will be healthy. Please, 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 did I mention please? Know that you are not immune to this virus. Okay. If 20% of the tests are coming back positive, chances are, even if you're not showing symptoms, you are harboring it right now, carrying it around with you, which means you could be potentially spreading it to others. So, with that positive sentiment, I said at the beginning of class, this paper is challenging, right? And I'm sure you have a lot of questions, and I thought how using a Pear Deck slide to kind of capture some of those questions might be helpful, but of course you can ask these questions out loud. But if you take a moment just to begin, you may not have any questions, and that's okay. What are you uncertain about? What are you wondering about? What are you curious about in the context of this Stinson et al. Paper? Okay, if you could just take a couple minutes here and jot down your thinking. Or oh, Alex, you should say that out loud. That's it.
I mean, just like a one word.
Can't? Yeah, whatever is important to you right now. Absolutely. Yeah, I mean, the paper had a lot of terminology with which you might be unfamiliar. Maybe you want to just say, what is this? What is this? Or maybe you're wondering about a bigger picture type of question. What does this have to do with what we're learning? I hope you made the connection, but maybe it's a little unclear. Okay. Yeah. It's okay to be lost. I'm hoping the story time will reveal chapter by chapter, page by page, how they're getting to an answer to the question, right? How they're making their claim. Yeah, I agree that the formatting of the graphs could have been better, but I'll say now, and I'll say again when we get to it, the labels on the bottom graph are the same as the labels for the top graph figure. You might ask, why didn't they just write them twice? I don't know.
That makes so much more sense.
Yeah. Yeah, it is only 4 pages, isn't it? It's pretty amazing. I guess 5 if you count the materials and methods. Yep, we're gonna go over the graphs. Definitely challenging. Could be hard to comprehend just based on what's going on in the world right now, right? How much of your brain is actually available to understand this stuff? I do hope the annotated paper that I created, that is my creation, for all its positives and negatives, I'm hoping was helpful. And folks at home, let me know with a thumbs up that you're ready to move forward. Or you can just say, go, or yes. Or you could even say thumbs up, I suppose. Elliot, I hope you're there. Ben, I know you're there. Thank you. Are we ready for story time?
Yeah.
Do you have your warm milk and teddy bear?
Why did they decide to call it Teddy Bear? Who is Teddy? Why does he own all these bears?
I don't know. Oh, Sarah, do you know? Sarah knows why it's teddy bear. Will you please share?
So yeah, it's from Teddy Roosevelt and that bear cub that he supposedly didn't kill, which is actually like sort of a myth story. Like the bear stuffed animal existed before it, but this old couple was like, hey, Teddy Roosevelt didn't shoot a bear cub and now he's popular. Let's name our bears after him and make money. And that's how that happened.
There you go. Thank you.
Wow,
I had no idea.
that's interesting.
Alex was on top of it too. Huh. All right. So then it was kind of ironic in the spring when little kids were going on a teddy bear hunt. You guys know about that? The little kids would like go around their neighborhoods. They were looking for teddy bears in the
My
window.
mom had to like hide some.
Yeah. Yeah. I don't know. My kids were doing it. They loved it. All right. Well, let's hop into this. Thank you for sharing your questions, your wonderings, your curiosities. I know it can be hard to say them out loud. So I'm on slide 139. Hope you can see it on your end as well. I thought I could just begin with kind of a summary of what's going on. So, when we think about even just the title of this paper, right? The mutualism that's taking place, right, out in the wild is between a fungus and these woody trees, right? These woody plants, woody bark, right? And this can be surprising to a lot of us because when we think of a fungus, we think of like a mushroom growing above ground. We don't think about what's below ground. And mushrooms aren't plants. They're actually more like animals than they are like plants. They are heterotrophs. So they don't have roots, but if we look underground, they have extensions underground that can actually make physical contact with the roots of plants. When I say physical contact, I'm not exaggerating. I mean they burrow their way into the root and make physical contact with the cells of the root. And they do that almost like a straw to siphon sugar away from the plant. They're drinking the sugar. You might say, that sounds like a parasite. I said, it does sound like a parasite at first glance, but this is not a parasitic relationship. This is a mutualistic relationship. Because that physical contact between the fungus and the roots of the plant also more efficiently delivers nutrients from the soil into the plant. So things like phosphates and nitrates, potassium. You ever seen the back of a fertilizer? There's an NPK ratio on the fertilizer. Different plants like different combinations of nutrients. N, nitrogen, P, phosphorus, K, potassium. It's almost like all that memorization in chemistry has reading significance. I actually heard a chemistry teacher say yesterday that in general chemistry, you don't have to memorize the polyatomic ions, but in honors chemistry, you do.
Yeah.
And I just jumped up and down and I said,
Yeah.
what, because that makes the class harder? Oh my goodness gracious. Sorry, aside. And you can publish that on YouTube too. I already am, I don't know why I'm saying that. All right.
Post all of this?
Not every single class, no. Because I figured that would be redundant if the same lesson now 8 times in a row is being published to YouTube. So I try to pick like one example. All right. Yeah, it's funky that a mushroom is not a plant. Remember, they're not green. They're not photosynthesizing. They're not producers. If anything, they're decomposers. So they are acquiring their nutrients from some other source. They are not using photosynthesis to make their own sugar. But it's weird. They're weird organisms. They almost bridge the gap between plants and animals. All right, so physical interactions between the fungus and the plant. Indeed, this is mutualistic in nature. I want to show you a picture of garlic mustard. Maybe you've seen it, maybe you haven't. You probably won't see it growing this time of year. We've already had a frost, but it might survive frost. But I know that I have flowers like this growing in my backyard. So maybe they'll get a little familiar to you. Garlic mustard is in Missouri. It is an invasive species, a non-native species to Missouri and lots of other Midwestern states. So everything that you're reading here has the potential to influence what's happening here at home. Okay? It's not some abstract thing that's happening in like Portugal. I don't know why I chose Portugal. No shade towards the Portuguese. Okay. So really what this paper is all about, right? Oops, sorry. Is, okay, what is the effect of this invasive non-native garlic mustard species on the mutualistic interaction between the fungus and the wood plant? Okay. And if we start story time, folks at home, can you still hear me? I'm pretty far away from the computer.
You're getting pretty far away, but we can still kind of hear you.
Okay, I'll move closer. Thank you for telling me. Isn't it cool to have that in the back? Come on, because that was Alex, right?
Yes, it was.
You know her voice. I don't know her voice. Thank you, Alex. All right, so I'm going to look at figure 1 first. Okay. And, you know, this is kind of a super simple experiment, right? They're going out into the world. They're identifying where the garlic mustard plant is growing and where it's not growing. Okay. And I'm not exaggerating. They're bringing their shovels. They're digging up the soil. They're bringing it back to their labs. They're not bringing the plants necessarily, but they're bringing the soil where the plants were growing or where they weren't growing. Okay. And to give you kind of a mental picture, I'll go back to the figure here in a second. Imagine hundreds upon hundreds of these little plant trays, right, that now have either invaded soil or uninvaded soil in them. And then into those little holes, right, really not holes, they're cells of that plant tray, go one of 3 types of seedlings: white ash, red maple, sugar maple. Okay. And then start the clock and come back 3 months later. If you are going into science as an advocate for instant gratification, please, 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 please rethink going into science. Sometimes these experiments take a long time. But they come back after 3 months, and now I'm back to figure 1, so go back. And again, the tricky part, right? The labels on the x-axis below are the same labels for the x-axis above. So if we look at first percent mycorrhizal colonization, mycorrhizae is another name for fungus. So fungal colonization. How is the colonization working for the plants that are growing in the soil that have been invaded by the garlic mustard? Not very well, right? In fact, we would argue that maybe except for the red maple, there's no fungal colonization of the roots of sugar maple or white ash plants. They're struggling, right? And if I compare that to the invaded soil, sorry, the uninvaded soil, is there a statistically significant difference between the 2 types of soil?
Yeah.
How do we know?
Air
Air bars don't overlap, right? Okay, that's an important piece of the puzzle. We can go a step further, right? We could say, now I'm in figure 1b, but I'm still on the left-hand side, invaded soil versus uninvaded soil. Percent increase in biomass, which is literally just a measurement of how much mass the plants have gained as they grow. How are the plants doing in the invaded soil? I mean, what do we got? 10% maybe increase in biomass? But compared to the plants that were growing in the uninvaded soil, holy heck, 250, 300% increase? Is that difference Statistically significant? Yeah. So the plants aren't doing well in the soil that had been invaded by the garlic mustard, right? Even though the garlic mustard plants are no longer present. Right? So if we move to the right-hand side of that figure where it says sterile invaded and sterile uninvaded, it's kind of a cool experiment, right? Same soil, but this time they're going to put it into a machine. We actually have the machine in the back. It's called an autoclave. And what those autoclaves do, what those machines do, is they heat samples, really anything, soil included, to about 120 degrees Celsius and about 15 pounds per square inch.
Did they do that when like what was in it when they did that?
So imagine that they had gone out into the world, into the forest, dug up the soil, brought it back to their labs, put it right in the machine. So the plants aren't in there yet. Okay? But I mean, think about that. 120 degrees Celsius, 15 psi. If I put you in there and you won't fit, but I'm always going to take volunteers. If I put you in there, you're not going to make it. The fungus is definitely not going to make it. So by sterilizing the soil, you're killing the fungus. And perhaps not surprisingly, is there any colonization of the fungus in the roots or of the roots in the sterilized soil? And it doesn't matter if it's invaded or uninvaded. You kill the fungus, there's nothing to do with the colonizing. And similarly, how are the plants doing in the sterilized soil in terms of their biomass increase?
Not good.
You can argue they're doing about as well as they were in the invaded soil. So this in figure 1, chapter 1, right? The authors are trying to tell us there's something in the soil, folks. There's something in the soil. And possibly whatever's in the soil is impacting the fungus. If I sterilize the soil, the effect that I see is similar to what would happen if I didn't sterilize the soil, but the garlic mustard had been present in that soil.
It almost acts in the sterile.
Something, yeah. Something like that. Are there other questions about figure 1 before we move forward? There's something in the soil. In figure 2, now they still want to know if there's something in the soil, but now they're not going to use field soil where the garlic mustard plants, while they're growing or weren't growing. Now they're going to use their own soil. So they go to the soil store, you know, Amazon, soil.com, and they set up an experiment that looks like this. Again, many, many trays, their own soil this time, but now they're going to grow white ash plants, red maple plants, sugar maple plants in that soil and wait 3 months. And then come back after 3 months, pull all those plants out, because what that's going to accomplish is condition the soil, right, with whatever it is coming out of those 3 types of plants. And let me add one more layer, right? The garlic mustard plants are in there as well, okay? So there's soil where the garlic mustard plants are growing, the red maple, the sugar maple, and the white ash plants. Pull all those plants out, keep the soil. Come back and now add new seedlings. Red maple, sugar maple, white ash, and see how those plants grow in the conditioned soil. Wait 3 more months, 6 months. Come back. Which type of conditioned soil, has the most significant effect on either colonization or increase in biomass? With that GM down below. GM stands for? Garlic mustard, right? So in the soil that was conditioned with the garlic mustard plants, right? You pull the garlic mustard plant out, you use the soil where those plants were growing, and then you add back seedlings of these 3 types of woody plants. something about the soil where the garlic mustard plants were growing that impairs the ability of the fungus to colonize the roots of the plants and inhibits the growth of these plants. So yeah, it's happening in nature, but they can verify that effect in their own laboratories.
How is the fungus getting into that soil?
The soil itself has fungus mixed in with it, so spores of the fungus. Actually really common for like potting soils or seed starting soils. There's fungus adding to it, fungus added to it as a microorganism. Very common. There's something in the soil, and the garlic mustard must be doing something to the soil. So figure 3 is my favorite. Because now, I kid you not, they literally make an extract of these 4 different types of plants. So imagine, you've got the plants, you throw them in a blender, grind them up with a little bit of water, and then add the extract to the soil where these different plants are growing. Red maple, sugar maple, white ash. Like, you could do this experiment, right? This is not beyond, like, you don't need a PhD to do these sorts of experiments, right? Sometimes we think scientists are so smart and have access to the most amazing resources. They're doing things that I can't possibly do. BS, you could do this. You just need a greenhouse. Oh wait, we have one upstairs that we're not really using right now. So they add the extract to the soil where these plants are growing. Which type of extract had the greatest effect? Still the garlic mustard. Folks, there's something in the soil. The garlic mustard are putting it there. And it's probably something chemical. In fact, it is something chemical. They're not at the level yet of being able to identify what that chemical is, but it's a chemical in the soil. So, sorry, I keep coming back here to make sure I check the chat. Thank you, Sarah, for adding GM. You're absolutely right. So, figure 4 is not my favorite figure in the whole wide world. So I'm going to quickly summarize it for you, just because it's the end of our story. I think it's pretty clear at this point that if these 3 types of plants are growing in the presence of garlic mustard, there's a negative effect, right? And that negative effect of the garlic mustard is occurring through the means of disrupting the mutualistic interaction between the fungus and these plants. So really all figure 4 is about is, okay, is this effect potentially damaging, not just to these 3 species of woody plants, but any plant within a living system that's dependent upon an interaction with the fungus? And what we see in both figure 4A and 4B is that, yeah, all those little dots that you see, those are different species of plants. Some of which are more dependent upon this mutualistic interaction between the fungus or with the fungus, and others that are less dependent. And the more dependent you are upon this interaction, the more likely you are to be affected if this interaction gets disrupted by plants like garlic mustard. That's what figure 4 is about. But here's why it matters. Let me pause there for a second. Does that bring any questions up in your mind? Is this remotely interesting? Okay. Let's see if I can make it super interesting. Let's see if I can, even through your face covering, get your jaw to drop a little bit. Because this is what garlic mustard looks like growing in a forest ecosystem. It can completely cover the forest floor. So as you look at a picture like this, is there light reaching the forest floor? Does it look like those plants, those garlic mustard plants are illuminated? Sure. Can you see the trunks of the woody trees? I tried to find a good image, but I'm not sure how good it is. Can you begin to see the branches and leaves that form the forest canopy? Not really, but we know they're there, right? Because there's something of providing some shade and blocking some of the light from reaching the forest floor. Is that fair? Well, folks, think about what the garlic mustard plants are doing. Not just in the short term, but over the long term. And I'm gonna argue that these darn plants are long-term planners. That they are exerting a behavior on the system, which is going to favor the system. Sorry, so it's going to change the system in a way that favors the survival of the garlic mustard over everything else. Because this is what tends to happen to forests that have been invaded by garlic mustard for a long period of time. What are you noticing?
Trees are dying.
Trees are dying. If I'm making it harder for established older trees to establish nutrients, right, to grow and develop over the course of their lives, they're probably going to be more likely to die as a consequence. We're seeing the evidence for that. Moreover, you could argue that, wait a minute, what about the new trees? The white ash, the red maple, the sugar maple, are they going to have a good chance to grow and develop in the system over time? So the older established trees are more likely to die. The new trees that would take their place don't have much of a chance to make it. How would the light conditions overhead change as the older trees die and newer trees never replace them?
Last shade.
There's less shade. Oh, think about that for a second. These damn plants are manipulating the system in a way to increase the amount of light that they receive, not in their own generation, but potentially for all of their descendants, several generations later. Is that a bottom-up or a top-down trophic cascade?
Bottom up.
They are producers. Sorry, Ben, I think it was you. Say it again.
Bottom up.
Yeah, it's bottom up. But this time, this bottom up cascade is not supporting species diversity, right? This is lab 7. It's going to decrease species diversity. And we don't even see it in pictures like this. Any of the species that are dependent upon those woody trees, oh crap, they're in trouble too. That calculation of the species diversity index, boom, 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 boom. Get ready for it to drop over the course of time. Okay. So invasive species, not great. So what do we do? We've got a couple minutes here. Not a lot, but a couple. What do we do?
Try to control them.
So how might we control them, Maya?
I mean, it's not always great, like chemicals.
Ideally, if there was a chemical that could kill them, that'd be great. Can't kill the other plants in the system, but if it killed the garlic mustard specifically, that would be a great idea. But even if that worked, if the garlic mustard plants are there, what problem exists?
Soil's different.
Oh crap, the soil! The soil's still contaminated. How do I get this chemical out of the soil? I don't really know. I can't dig it all out and replace it with new soil. I would disrupt all of the interactions and the root systems of those established plants. That's not going to work.
Are there any other organisms which can kind of like solve this one?
If you could introduce a new organism, a new species, that would be interesting, right? But you better make sure that it's native and not invasive, because you could be right back to square one in terms of disrupting the system. But it's not, it's a great idea. You wanna know what we are doing right now? And it's not great. We're hiring high school students and college students to go into these forests and pull out the garlic mustard and burn it. Or we're creating controlled burns in the forest to kill the garlic mustard. Doesn't solve the soil problem, but at least it helps reduce the number of garlic mustard plants that are in the system. Short story long, these species don't belong there. They're wreaking havoc. The solution is make sure they never get there in the first place. So, if you see a plant that has flowers like the one that you saw in that image, rip it out. You probably don't have to burn it, but rip it out. Because you certainly don't want it to prosper in
Put
the
it in
future.
compost and it would go back in.
Especially if it's and it's seeding, if it's formed seeds, that would be problematic. If it's just the flower, well, you could argue, well, yeah, you have to begin to wonder, you kill the plant by pulling out the entire plant, is it still able to produce the chemical that would make its way into your garden soil, right, your garden compost. Well, I hope this story time was helpful. Kill them, says Alex. Alex, you should have said that with like an evil, maniacal voice, especially now that you're on speaker. We could have heard you. All right, last question before you take off. Again, you got a text a couple days ago. The next summative assessment, still take home, is live for you. What do you need between now and that due date, including more time, if necessary, to be as successful as you possibly can?
You should do.
I have the due date set for Monday, right now, with no other homework between now and Monday. But if you think there's something else that I can do that would be helpful, I would appreciate you sharing your thoughts with me. If you don't have anything, that's okay too. What is it in the Muppets, the new Muppets movie? Maniacal laugh. Maniacal laugh. Nobody? Anybody?
We do number 7. I was in the list.
I mean, really, it's all about the practice, right? Like even now, like for additional practice problems, the purpose of that lab was to serve as a practice problem, right? If you think you understand the role of bottom-up and top-down trophic cascades and how they connect to those science ideas, no, you don't need to do
Yeah,
work
I feel
with it.
pretty good about it. I think I just
Yeah, you weren't-
misunderstood the direction.
Yeah, I'm just trying to create examples for people, not necessarily like this is what you should do or need to do.
Thank you so much.
Of course.